for a real charismatic species, that is the sea urchin. I will be talking about the molecular characterization of the opsin-4 gene in Paracentrotus lividus sea urchin. First, a little bit of a background story on the pioneer and the inspiration behind this project, which is this guy here, is the Paracentrotus purpuratus sea urchin, which is the Pacific sea urchin. It has been used for like almost a century for many developmental studies, including gene regulatory networks, gene functioning, cell type, and nowadays, since the publication of his genome in 2006, omics and even gene therapy techniques have been developed. However, in my project, I used the sea urchin Paracentrotus lividus, which is the local sea urchin in the Mediterranean and Atlantic. And the reason behind this is due to its faster development of its transparent luteus larva, like we see there floating around. And also because it's a local species, and it makes kind of a little bit more sense to not have a Pacific species all the way from California to Europe every time you need to use them. So moving on to the objectives of my thesis project, they're the following. First, developing a CRISPR-Cas9 protocol for the knockout of the opsin-4 gene in P. lividus. Culturing Paracentrotus lividus larvae through to metamorphosis. So first they have a larval stage, and then they have to metamorphose to become a juvenile and then grow to adulthood. And then finally, the molecular characterization of this gene by means of immunohistochemistry. So it's very wordy and complicated, so let's just break it down a little bit. Photoreception. It is a key thing key characteristic in so many processes, in so many animals, that drives evolution in so many different ways. And in a few words, how photoreception works in general, you have a light signal, and this signal is captured through the eyes. In the eyes, we have photoreceptor cells that have genes like opsins, which is the gene that one of the genes that I'm interested in. And in the eye, these photoreceptors translate the signal that is a photon into a neuronal signal, and then the brain has that information and does something with it. And however, how does the urchin see with no central nervous system and no real eyes? So is it just one large eye in the entire animal that then they suddenly are able to see through that one eye, or is it just tiny little eyes all around the sea urchin? So it's kind of, it was found out in studies in Paracentrotus purpuratus, sorry about that, that it is actually a combination of both. So it is found out that throughout the entire sea urchin, there is photoreceptor cells in all the appendages and epidermis. And in a compound eye-like formation, all of these photoreceptors, which are not organs, they combine the information and act as one huge eye. The sea urchin is like one huge eye walking around. And yeah, it does walk. They're not seesaw. And this is the opsin family. It has already been studied a bit more since the publication of the genome. And the type of it is a huge family of different opsins. And the one that I'm interested in is the rhabdomeric opsin in the corner there. It's opsin-4 because it is the most strongly expressed opsin in the tube feet of sea urchins. So first, a little background on CRISPR-Cas9 for the ones who haven't heard of this infamous tool. It's actually not very complicated in theory. You have a Cas9 molecule, and then you have the gRNA, which is a guide RNA that you design yourself. And this guide RNA is specific to whatever gene that you're looking for in your genome, in your organism of interest. And in combination, the gRNA and Cas9 complex form the CRISPR-Cas9 complex, and that goes around your genome and looks for the PAM, which is the protospacer adjacent motif. And it's just a tiny sequence that the Cas9 recognizes. And once that happens, the gRNA opens up the heteroduplex, and the Cas9 is activated, and a double-strand break takes place. That double-strand break happens, and then is fixed through non-homologous end joining, which just means that the two bits on either side are ligated. And because this is kind of like a quick, dirty fix, a mutation is generated and a gene the gene is completely knocked out, if this happens in the coding region, of course. And the process was kind of equally simple, in theory, again. First, you design and synthesize the gRNAs. In my case, I had 4, 1 for the exon 1 and 3 for exon 2 in this particular gene. And then you extract the gametes from the adults. You don't kill them, by the way. And then you fertilize them, and the microinjection occurs soon after that. And then you grow them, and then you extract DNA, and through Sanger sequencing, you can tell whether the mutation took place or not. Moving on to immunohistochemistry is actually not that complicated to even do or understand. You basically have 2 sets of antibodies. The primary antibody is the one that is designed particular to your gene of interest. So in this case, we used actually one that was already developed for S. purpuratus, that is anti-opsin 4. And also a positive control is anti-acylated alpha-tubulin, which is just a marker for cilia and then neurons in later stages. And to that antibody, a secondary antibody, it recognizes it through like a common basically binding site. And this secondary antibody is the one that has the fluorophore. And those antibodies are just bought. They're manufactured worldwide, and you just buy them. And these guys are then the ones that you can see through a confocal microscope, which is just a very fancy microscope. And you can see basically individual cells. It's very stunning. We'll see some stuff now. First results, CRISPR-Cas9, unfortunately still in progress. We didn't manage to get any mutations for now. However, I haven't lost my hope, and I'm going to be there for another year. So I will be redesigning gRNAs, most importantly, and hopefully maybe also doing some positive controls with a more representative gene, like that has a phenotype, because obviously you can't see the eyes, they don't actually have them. And testing these gRNAs more thoroughly, because I did a cocktail of gRNAs, and I didn't test them individually. My intention is to test them individually, and then as a cocktail to increase the mutation. And then another idea nowadays, Cas9 molecules are manufactured also very worldwide, and is the idea of using a fluorescent one to be able to tag, to actually pick out the larvae that are actually carrying the mRNA that have managed to translate it accordingly. So that way I can identify it if they at least have the Cas9, so I can target them and be like, actually it's working or it's not working. And then trying alternative assays to detect mutations. I used maybe a not very thorough method because I didn't have that much time, but the idea is to try some new methods that involve all the tools to be able to have a larger like sweep of like all of the micro-injected larvae that I have. And then, as everything, is just to keep trying. Results with culturing the sea urchin larvae. At first, we had some very ugly guys, like those ones with spicules going wrong and like skeletons looking funny. And that was mainly due to adjusting the feeding rates. And the chosen algae was Dunaliella terticolecta after like a lot of working around it, essentially. But however, we did manage to get the full cycle, and it's this very beautiful image from an unfertilized egg through all the cell divisions, and then gastrulation, and then all the Pluteus larva stages from the 2-arm, 4-arm, 6-arm, and the 2 last ones are the 8-arm larva. Take a look at the last one, which is where you can find the rudiment. The rudiment is the section of the larva that then gives rise to the juvenile. So basically, the juvenile eats the larva and becomes juvenile. So as you see this guy here, they start eating and eating, and then the rudiment then develops into the baby sea urchin, essentially. And from A to D, we see the transition between the metamorphosing juvenile, and here in C, we can see the 5 primary podia, which are the first 5 tube feet that a sea urchin has for the first time. Okay, so now the immuno-detection of opsin 4. This was the first time done in this species, and we did manage to get what we expected from previous literature, which is you can't really see the ones on the other sides, but you can really see the one on the top, which is one of the basal positions of the primary tube feet. So at the basal position of primary tube feet is where we can find opsin-4 signal. And most importantly, we can kind of see the link between the neuronal, a neuron and the photoreceptor cells, which I think is very beautiful. And also, again, that is the acetylated tubulin in green. That is the positive control that is tagging both neurons and cilia in this species, in this particular stage, which is the 8-armed Pluteus, which is the one with the big rudiment at this stage. And this one is a wild-type organism, which means that it doesn't have CRISPR injected. But this one here does, which confirms again that CRISPR did not work, because we can see the expression of opsin-4 again. And again, here you can see it a little bit better. This one it doesn't have the opsin-4 because it's just, I removed that layer. But in this one, we have only acetylated tubulin and opsin-4. And we can see more or less the 5 locations of the 5 primary podia of a juvenile. And this one is a microinjected. And again, those two here are also microinjected. 8-armed Pluteus, just a close-up of the 2 little cell cluster that has 2 cell clusters of the photoreceptor cells at the basal position of the primary tube feet. And then this is a juvenile tube foot that you see the 2 cluster at the base and then actually at the rim of the tube foot. And because I wasn't able to grow any wild-type juveniles, I took the adult tube feet and did immunohistochemistry on this as well. And I think it's a very stunning picture. They have a segmented tube foot that you can't really see here, but again, on the rim of the tube foot disc, you can see all the photoreceptors and the elongated shape that then joins with the neuronal, in the neurons for phototransduction. Final thoughts on my project. I managed to make the first molecular characterization of this gene in this particular species, Paracentrotus lividus. We had a successful culturing of the larva through to all the way to metamorphosis and after metamorphosis. And then we took some steps forward towards achieving CRISPR-Cas9 protocol, which has only been implemented in the other species so far, and definitely not in this gene. So that's in progress. And first, I would like to acknowledge my supervisor, who has been great, my lab partners, Daniela, Pericles, and Maria, and my friends and family, and obviously Embassy, Erasmus+, Stazione Zoologica. And I would like to thank all of you for listening.